Hello and welcome to the Microbiome Medics Podcast. I'm Dr Shiv McCormack.
And I'm Dr Sheena Fraser, and we're your co-hosts.
We are both GPs and lifestyle medics with a shared passion for microbiome science, and we have spent the last 5 years deep diving into the world of the gut microbiome.
We've been analysing the evidence, appraising the studies, speaking to the experts, and writing the Gut Microbiome for Clinicians e-learning course for the BSLM Learning Academy.
We have discovered that the gut and other human microbiomes are pivotal to every aspect of your health and physiology, and the pillars of lifestyle medicine hold the key.
So in these Microbiome Medics podcasts, we'll be covering the basics, bringing you the latest research and talking to the experts on all matters microbiome.
We'll be translating the evidence and packaging it into actionable, bite-sized chunks so that you can harness the power of the microbiome to improve your own health and that of your patients.
Hello, and welcome to this month's edition of the Microbiome Medics Podcast. So, every 2 minutes in the UK, somebody's diagnosed with cancer, and sadly, every 4 minutes, somebody dies of cancer. 1 in 2 of us is going to develop cancer in our lifetime. So, given how serious a topic this is, we plan to spend about 3 episodes looking into the relationship between the microbiomes and cancer development, and cancer treatment and survival. In today's episode, we're going to be discussing the role of the gut microbiome in cancer development. We're going to be looking at the hallmarks of cancer cells, what it takes to make them successful. Then we'll be considering the role of the gut microbiome in cancer prevention and development. And Shiv's got a fantastic paper to discuss on the role of diet and the gut microbiota in colon cancer development. So let's get started. Hi, Shiv. How are you?
Hi, I am really good. I'm excited about this topic. It's incredibly relevant, incredibly important, and there'll be lots of people out there who are concerned about cancer for themselves or who know someone who has cancer or be struggling with that diagnosis themselves. So I think it's really, really important.
Yeah, fantastic. So tell us what you've been looking at today, because I think you're going to kick it off about the cancer cells and what they are and, how they're so successful.
Yeah, so people worry about cancer. It's one of the biggest things that people express concerns about when they come and see you and I in the GP surgery. And it's typical in a GP consultation that someone will come in with a lump or a lesion. It'll be palpable or visible to the naked eye. So, for example, a breast lump or a testicular lump or a mole that looks a bit odd. And they'll obviously be concerned and want you to tell them, is it cancer or isn't it? So often this involves a referral to the rapid access clinic, further tests are done, and the patient's hoping that there will be a yes-no answer here. But the idea that cancer starts on the day of a diagnosis is clearly incorrect, and the idea that a doctor or any number of blood tests or scans can tell you whether you do or don't have cancer is also incorrect. So I just want us to really slightly alter our perspectives on cancer in 2 ways, and if you bear with me, this is really important for understanding how the gut microbiome relates to cancer. So there's 2 ways, Sheena, I want us to think about these perspectives. One is in terms of time, and the second is in terms of size. So if we look at the time perspective, so there's a long lag time from initiation to cancer diagnosis. It can be years. So for example, with the adenoma to carcinoma sequence in colorectal cancer, there's a long, many years lag time between the initiation and the actual cancer diagnosis. And secondly, I just want us to rethink size perspectives. So lesions that patients show us that are visible to the naked eye or palpable, they've been present for a long time. So I want us to use our imagination skills just to think small, think tiny, shrink right down to the size of a single cell. And I want us to think about what skills I would need, for example, to become a successful cancer cell. So cancer is a disease of the DNA. So a cancer cell needs genetic DNA damage and mutation. And good sources of damage are environmental sources like, we know about UV light and melanoma. We know about smoking and lung cancer. Other environmental causes are alcohol, pollution, red and processed meat, and low-grade chronic inflammation. So those are environmental causes of DNA damage. It's important to remember that a lot of damage occurs unavoidably during your normal biology. So just the byproducts of energy production in your cells. So not a lot you can do about this. And then there's this sense of DNA damage accumulating over a long period of time. So a successful cancer cell is able to ignore growth suppression signals, and it can activate proliferational signals to make it grow. It's able to hide from and avoid destruction by the host immune system. A successful cancer cell can resist cell death and become immortal, essentially. It's able to access blood supply to receive nutrients and move around the body. A successful cancer cell thrives in a pro-inflammatory environment, and it's able to replicate, grow, invade, and metastasize. So all these things that I've just described come under something called the hallmarks of cancer. So this is a well-accepted conceptual framework. It was by two scientists called Hanahan and Weinberg, first proposed in 2000. And it's a way of simplifying the vast complexity of cancers into these key principles that are shared by most cancer types. Now, these hallmarks are added to as the science evolves. And before you ask what's this got to do with microbiomes, in 2022, a new proposal suggested that microbiomes should be added to this framework because they're so important in every aspect of cancer. Now, I know you're probably bored of hearing me talk about this, but by cell count, we are 50% microbial cells and 50% human cells. So the human cells is about 30 trillion, microbial cells about equal numbers. So let's just go back to the human cells. And we just think about one of these 30 trillion cells. Each cell has tens of thousands of DNA insults every day. So you can see that our body has to cope with a lot. And the cancer results from this DNA damage over a long period of time. So rather than talking about why we get cancer, I want to turn it around and say, well, with all this information and all the damage that each of our cells is getting every day, maybe we should ask why we don't all get lots more cancers more of the time. I think that's really important. I think it's a really positive way of looking at things. And the answer is because our biology is brilliant. So we have these inbuilt safety systems that do three things. They prevent this DNA damage when they can. But if this DNA damage occurs, they then lots of safety systems to repair this DNA damage. And even if cells break through, we've got an incredible immune system which is constantly patrolling, looking out for dodgy cells and knocking them out. So the gut microbiome in particular influences all these inbuilt safety systems. So genomic mutation, the immune system, modulation of signaling. So the gut microbiome is incredible, this complex dynamic ecosystem that can be tumour protective or tumour promoting by many different pathways, depending on which microbes are present, what quantities they're present in, and what they're doing, what their function is, and what their metabolic output is. So Sheena, you can see it's pretty complex, but there is an intricate connection between the gut microbiome in particular and all these safety systems that prevent us getting cancer. So I'm going to hand it over to you, Sheena, now to tell us a bit more about the gut microbiome and the role it plays in cancer development.
Yeah, thanks, Shiv. It is amazing, actually, just how these safety systems are all in place there the whole time, and just how many times we're sort of so close to developing cancer, but our body's immune system kicks in. It's absolutely incredible. So yeah, I mean, let's move on to the gut microbiome itself and what role it plays. And actually, it plays pretty incredible role in our immune system. We know that 70% of our immune system is within our gut microbiome. And so, of course, it's playing an essential role at all times in our anti-cancer strategies. We've already discovered in previous podcasts how important it is at the start of our life to develop a really healthy gut microbiome. So an unhealthy balance of more harmful strains of bacteria within the gut microbiota is associated with a leaky gut. It's associated with increased inflammation, which can be a precursor to cancer development. It is associated with obesity. Again, people with obesity are more prone to cancers. It can be associated with increased chronic disease. And we know people with more chronic disease, again, are more prone to cancers. So we know that development of our gut microbiome is really important. But we also know that people with chronic disease and obesity get these cancers more readily because of lots of other mechanisms going on in their gut microbiome. There's also, it's important to realize that we already know that there are some microorganisms that play a direct role in causing cancers. So, for example, H. pylori can trigger stomach cancer. Viruses can cause cancers like HPV virus, human papillomavirus. They're associated with cervical and tonsillar cancers. Hepatitis B and C are viruses that are associated with liver cancers. But the story with these viruses isn't straightforward either, because H. pylori can actually protect us from esophageal cancer, but give us a higher risk of stomach cancer. So it's there's a sort of mixed message with these organisms at times. So when it comes to individual organisms, there can be risks associated with individual organisms. But what's really interesting is when we look at microbiomes as a whole, when we look at the whole ecosystem, and some of these ecosystems can actually promote cancer development, whilst others can inhibit cancer development. So if we've got a very, very healthy gut microbiome, then you'll have lots of really beneficial microorganisms in there that will work very hard at our cancer surveillance systems and help to reduce cancer development. Meanwhile, a sort of very unhealthy microbiome with lots of harmful bacteria may not be quite as effective at tumor surveillance. And actually, these pathogenic or more harmful organisms may be also producing toxins more readily that are associated with the development of cancers. For example, some bacteria produce sulfur gases, which are associated with promoting colorectal cancers and methane as well. And so, these sort of toxins that they can produce are also associated with more damage. We know that lipopolysaccharide, which is a byproduct of a sort of membrane of the wall of a harmful bacteria, and when it's broken down, it can be very toxic within the bloodstream and create a lot of inflammation. And again, high levels of lipopolysaccharide in those individuals with too many harmful bacteria in their gut microbiome can be very pro-inflammatory and harmful and increase our likelihood of cancer development. So if we have this, this imbalance there, then that can also mean that we produce fewer short-chain fatty acids. And we know from previous podcasts that we've talked about short-chain fatty acids and how important that they are in terms of developing our immune system. So they are important in terms of stimulating our very anti-inflammatory cytokine developments. So if we don't have enough short-chain fatty acids, if we don't have enough beneficial bacteria to produce them, then again, we can be missing that important immune surveillance. Okay, so certain bacteria can have direct cancer-promoting properties. For example, E. coli, which is a common bacteria that lives in our gut, but can also be associated with quite severe infections and things. But it can also produce a toxin called colubacin, which causes breakages in DNA, and that's been implicated in colorectal and breast cancer. In addition, there's a bacteria called Strep pyogenes, which also seems to increase our systemic estrogen. So when the estrogen is metabolized, it goes into the gut, and then some bacteria like Strep pyogenes can actually metabolize it again into a more active form and push it back into the circulation. So that can raise the levels of estrogen in our bloodstream, and that heightens our breast cancer risk. So we all have these bacteria in our microbiomes, and in small quantities, these bacteria seem fairly harmless and actually probably do a lot for our health in general. But when these numbers of these bacteria get up into higher levels, that's when the harm can start. We talked about leaky gut in a previous episode, but when bacteria and their toxins leak through the gut barrier, they can create inflammation elsewhere in the body. This inflammation can be a precursor for disease, as I've mentioned, and indeed cancers themselves have microbiomes And these microbiomes are almost certainly developed from gut bacteria that leak through this membrane and travel to the site of the tumor. So we've discovered that, for example, pancreatic tumors have high levels of gut bacteria within them, as do breast cancers and many other cancers. In fact, there's very interesting research going on looking at the microbiomes of cancers, which is really interesting. So, in contrast, our bacteria can work to help us prevent cancer. For example, Strep thermophilus can produce an antioxidant metabolite. Prevotella produces lots of short-chain fatty acids, which are very anti-inflammatory, anti-cancer, and can promote really acidic conditions in the gut, which will directly kill cancer cells, which is interesting. And many species of lactobacilli appear to inhibit cancer growth in animal models. So beneficial bacteria can really help to protect our gut wall. It can aid the production of mucus, which is a further defense of our gut wall, and it can inhibit harmful strains from overgrowing and releasing toxins. So goodness, there's an awful lot of ways in which our gut microbiome can either help to promote or inhibit cancer development. It's also worth noting that it's not just diet that will alter our gut microbiome. We also know that, of course, other lifestyle factors like sleep, stress, exposure to harmful substances, and exposure to nature and relationships all have an impact on the health of our gut microbiome. So it's important to know that, your lifestyle is very much, key to lowering your risk. Okay. So there are lots of studies that have shown that there's a link between a healthy diet and a lower risk of cancers. And diets high in fruits and vegetables with fibers and polyphenols, which really help to encourage the growth of these very beneficial bacteria in our gut, and increase our short-chain fatty acid production, can lower our cancer risk. And certainly the Mediterranean diet has lots and lots of research into that and reducing our cancer risk. Okay, so in addition, fermented foods can help to reduce the risk of some cancers, and probiotics, these beneficial bacteria, can be useful in cancer prevention and treatment, although the same probiotics can have different impacts on different people depending on individual gut microbiomes. So I think I'm going to stop there because that was an awful lot to say on the gut microbiome and cancer. It's beginning to feel like a bit of a marathon, but you can maybe understand a bit more now why we think that the gut microbiome is key to cancer prevention and cancer causing. So Shiv, you've been looking into a paper which kind of highlights this a little bit more. So perhaps you could move on to that and tell us about what you've been looking at this week.
Yeah, I will, Sheena, but I just want to pick up on a couple of points because it's incredible, the stuff you've been talking about. There's two things that I just want to focus on. First, it's this, if you're anything like me, if listeners are anything like me, you just want to know, tell me what the good bugs are, tell me what the bad bugs are, and then I know where I am. And unfortunately, it's not like that. You really have to think about your gut microbiome as a complex, dynamic ecosystem. And in the same ways, you couldn't look at an Amazonian rainforest and just say, list me 5 plants I need, or list me 5 environmental factors that I need to keep this area healthy. It's exactly the same with the ecosystems in our bodies and in our gut. That things that can be very beneficial in certain balances, if they overgrow or if they don't have other microbes keeping them in check, they can then become potentially toxic or carcinogenic. So it's not, that one bacteria is going to be labeled as good or bad. It's all to do with the balance. So these dynamic ecosystems can be cancer protective or cancer promoting by numerous interconnecting pathways depending on many things, including the environmental factors, your genes, what you're eating, the microbes that are present and what quantities they're present in, whether you've had antibiotics, what other drugs you're on, your other lifestyle factors like sleep and stress, and all these things are interconnecting in a massive network. And the second thing that really struck me about what you said, Sheena, bearing in mind that we know that microbiomes are everywhere, on every surface of the planet, on trees, in deep oceans, on high mountains, even on a grain of sand. So it shouldn't really surprise us that the cancer within us has its own microbiome. And it's pretty amazing to think of that a cancer, a lump or a breast lump has its own microbiome. And this is something that scientists are really interested in, because they're trying to use this information to see if they can alter chemotherapy or treatment protocols to use this in our favour. So that's something that's kind of really exciting going forward. So anyway, I just thought I'd say that. But going back to this paper, Sheena, I hope you're sitting down, because this paper is from 2015. Now, if anyone listening is wondering what I'm going on about, Sheena is allergic to any papers older than about 12 months. And she's kind of really cutting edge, always on PubMed, looking up the latest papers. Whereas I'm a bit more old school. I like these landmark papers from way back, 20, 25 years, those old original researchers who started this whole thing rolling. I just feel that you can learn so much from those classic landmark papers. So this is my favourite. One of my favourites, and Sheena knows this because I always talk about it, Fibre, Fat and Cancer Risk in African Americans and Rural Africans. It's by Stephen O'Keefe. It's an international project. And actually, lest you think that everything interesting in the microbiome happens in America, there's also Imperial College in the UK was really integral to this paper as well. And Sheena, I've become such a gut microbiome nerd. I can't read. I used to love reading trashy magazines just to relax, but I can't read a trashy magazine anymore. I'd much rather just kind of, if I'm feeling a bit stressed about, reading through all these papers, I will go back to this paper and just read it for pleasure. Now, I think that's slightly worrying. I think I need to see someone. But anyway, I love it. It's like a favourite song you listen to again and again. So that's how much I like it. So before we go into the paper, and I think you're going to fall in love with it too, it's about colorectal cancer risk. So we're just going to give you a bit of background there. So colorectal cancer, third most prevalent cancer globally, second leading cause of cancer death. But the amazing thing about it is how widely the rates vary by where you live. I mean, it's astonishing, really. So we've got really high rates in high-income countries like the UK and America. And this really contrasts starkly when you compare it with the annual colorectal cancer rates in rural African countries in particular. So the rural Africans, it's almost unheard of. There's less than 5 cases per 100,000 per year. Now, when you compare that to African Americans, so people who may generations ago have moved to Africa, it's 65 per 100,000. So that's about kind of 114, I think. So why is this? Why if you live in rural Africa, is it, unheard of or very rare? And then if you move to America from rural Africa, within 1 or 2 generations, you've then got these really high rates. Now, they know from migrant studies that it takes about 1 or 2 generations to assume the high rates of your host country. So it's not genes. So there's 2 potential mechanisms for the differences in diet-associated cancer risk. So what they've looked at is the protective effects of dietary fibre. So we know this dietary fibre is fermented by your gut microbiome to produce beneficial butyrate, which in general is anti-inflammatory and anti-tumour. And number 2, high levels of animal fat stimulate liver bile acid synthesis. Now, just a quick recap. We know that when you eat anything fatty, particularly animal fats, it causes your gallbladders to squirt out bile acids, which help you break down and digest these fats for absorption and digestion. So the gut bacteria capable of a bile acid conjugation can transform these bile acids that you produce, called primary bile acids, into secondary bile acids. Now, some of them can be beneficial, some of them can be harmless, but some of them can be potentially carcinogenic, and that is the problem. So with all that in mind, let's go on to the paper. So the people who wrote this paper were wondering about this differential in colorectal cancer risk between African Americans and rural Africans, and they wanted to investigate the influence of diet. So they looked at the differences in diet. So we know if you look at a typical African-American diet. So if you think of the USA, you can just imagine McDonald's, burgers, chips, all the usual, lots of high fat, high sugar, ultra-processed food. So their animal protein intake, unsurprisingly, is 2 to 3 times higher. They've got lots of ultra-processed foods, very low complex carbs, very low fibre. Whereas the rural Africans are having this beautiful, high fibre, complex carbs, loads of veg, really, really healthy diet. So we also, number 2, that if you change your diet radically, it can alter your gut microbiome composition within 2 days, 48 hours. So it's really, really quick. But when you go back to your terrible diet again, then it just swings back quickly to the, an unhappy, dysbiotic gut microbiome. So what they did with this study was they took 40 people, 40 people. So they were between 40 and 65, 20 African Americans and 20 rural Africans. Now, just as an aside, the rural African people came from South Africa, from the KwaZulu region. That's near Durban. And it's really important because there is a lot of research being done into gut microbiomes in indigenous populations and rural populations. And I was really pleased to sort of read that there's quite a lot of sort of advice to community leaders and specialists in minority studies to help people do this in a sort of dignified and respectful way, so that we're not just kind of getting research and not doing it in a proper way. So informed consent was got from these people, and the adverts were placed in community centres, and the volunteers, as I understand, were appropriately compensated as well. So that's kind of really important, especially important because they were asking these rural Africans to do 2 things. One is consume our Western diet, which is carcinogenic and addictive. And secondly, they were doing some invasive tests as well, like a colonoscopy. So it's kind of, it's really important that it was done in a correct way. So at the start of the trial, before they did the actual assessment, they did baseline tests. So we've talked about the differences between the diet, the African Americans having much higher animal protein and low fibre, and the rural African having essentially a whole food plant-based diet. So they did tests, they did a colonoscopy in all 40 people. The African Americans had really high rates of polyps, and we know that colonic polyps are precursors potentially to colon cancer. And I think it was about 40%, which is incredible, had 1 or more polyps. Rural Africans, 0 polyps. I mean, that's an incredible difference already. Yeah, I mean, it's amazing. Then they did stool analysis to look at 2 things, Sheena. One was the gut microbiome composition. So that's just the list of bugs that were there. But in a way, we don't want to know just what bugs are there. More importantly, what are they doing? So that's looking at the metabolites, the chemicals that those bugs are producing. So they did a stool analysis for that as well. And the difference was really, really marked. The African Americans had far more potentially pathogenic bacteria, which were capable of producing these secondary toxic bile acids that we know can be potentially carcinogenic. And number 2, there was a far less butyrate production. So the butyrate levels in the stool were really low. Comparing this to the rural Africans, they had really high levels of butyrate. Not surprising. They're having whole food, plant-based diet. Their gut microbiome is very happy. It's got lots of nutrients to produce these brilliant beneficial butyrates, and they had very low secondary bile acids. So they compared the colorectal cancer risk profile. In the African Americans, this profile risk was high. The rural Africans had a very low risk profile. So then we've got this 2-week diet swap. So the African Americans get to eat this wonderful rural African food. So I don't know if you've ever tried much African food, Sheena, but one of the main things they eat is something called, chocolata, which is a kind of looks like a vegetable bean chili, kind of really whole food, plant-based heaven. And they serve it with something called mealie pap, which is kind of a corn-based, I suppose it looks like porridge, and it looks a bit like polenta. But in case anyone hasn't tried this, we'll add I'll get a recipe, Sheena, and add it to the show notes, because it's kind of would be fun if we try this food, because it's so good for you, and it's just like, your gut microbiome, heaven. That's what they would order in a restaurant if they could. So I'm sure they were all delighted, the African Americans. They were eating all this incredibly, healthy and enjoyable food. But the rural Africans, I can't imagine what they were thinking when they were faced with what presumably was kind of pizza, chips, burgers, all the rest of it. So, they got all this stuff. Hopefully they hated it, but you never know. It's quite addictive, and we know that high sugar, high fat kind of gets your brain chemicals going. So, but hopefully they didn't carry on with it afterwards. So they only did this diet swap for 2 weeks. It really wasn't very long, and I would have thought that's not going to be enough time to make any difference. But actually, the difference was incredible. So they repeated the tests after just 2 weeks. So first of all, colonoscopy. So that's not going to change. The polyps aren't going to change. But what did change is something called the mucosal proliferation rate. So those are the kind of the excitability of the cells in the colon, in the mucosa. If they've got high proliferation rates, it makes it much more pro-carcinogenic. And so what happened after 2 weeks is the rural Africans were showing up much higher rates of mucosal proliferation, and the African Americans, their rates had gone down. The rural Africans now have really high levels of these toxic secondary bile acids, which are pro-carcinogenic, and really low rates of the butyrate. So completely different. The African Americans, their gut microbiome are now happy, and they're producing a lot more butyrates. Their butyrate levels have shot right up, and their secondary bile acid, the toxic type, have gone right down. So now, in just 2 weeks, Sheena, the rural Africans have a high colorectal cancer risk profile, and the African Americans have a low cancer risk profile. And I kind of find this amazing. So dramatic change in just 2 weeks. But I also find it really, I suppose, positive, because it just shows what you can do in such a short time. So yeah, I don't know what your thoughts are about it.
Yeah, absolutely. I mean, it's incredible the impact of the Western diet, isn't it? I mean, going back to that particular diet that the rural Africans were eating, I mean, the quantity of fiber was huge in comparison to the quantity of fiber in an average Western diet. And then, of course, what about all the preservatives and the additives and the things that we're adding into our foods now, into ultra processed foods, which we know are potentially now carcinogenic. And that's adding a whole new dimension to Western food. It's not just the high fat and the high sugar. It's all this other stuff as well. In fact, I've just before I went to record this podcast, I was actually reading a really new study that's just come out. It's called Toxicological and Pharmacokinetic Properties of Sucralose 6-Acetate and its parent sucralose in vitro screening assays. Now, sounds really, really complex, but this article is really fascinating, because basically it looked at sucralose and the impact that it has on the gut microbiome, and the toxicity that it has in our gut. And sucralose obviously is a very commonly used sweetener added to many, many different processed food products, including, the average soft drink that we consume, and lots of other processed foods alongside. And of course, with the rise in sugar tax, it's actually very difficult to find a sweetened drink that doesn't contain sucralose or another type of a sweetener. And interestingly, what this new study has shown is that sucralose actually alters genes, so it's genotoxic, and it's actually it can induce more cancer genes, so it can actually promote cancer genes, as well as increasing leaky gut as well. So it damages the gut wall, and these are all effects that the food industry, I don't know if they were aware of these effects, but they've certainly claimed in the past that they didn't have any of these effects. So this is very new information on sweeteners, and I think vital information on sweeteners. But that's just another thing to add into the sort of Western food story at the moment, that, it's not just about high fat, high sugar. There's all these other dimensions within Western foods that will add to the increasing cancer risk. And I'll put a link to that paper at the end of the show notes as well, because I think that that's going to be quite a vital paper in terms of looking at food safety going forward.
I suppose it's really interesting because, of course, when all these additives and sweeteners were brought out and were generally recognised as safe, they had this GRAS label on them that it was okay, they were safe. The gut microbiome wasn't a big thing then. When it started, it wasn't even heard of back then. So, they haven't been tested for their effects on the gut microbiome. And we just know that all these emulsifiers in particular are really toxic to our gut microbiome. So even though you think you're eating something, from a shop, which is kind of, plant-based, microwave, whatever, it's got so many emulsifiers in that it's just going to negate anything good that might be in it, even if it's a vegetarian chili or whatever. If it's covered in additives and emulsifiers, it's just going to be really toxic to your gut microbiome. So there's this thing about whole food plant-based. If you go into the section that's plant-based, you're really, if it's in packs and you need to microwave it, it's not really whole food. And it's obviously much better to just do something really simple like this chocolata, a recipe which I'll stick in, which is basically just chopping up a few veg, a bit of veg, putting in a few beans, and really cheap, really effective, really good for your gut microbiome. And also the other thing, Sheena, was just this thing I've got to be in my bonnet about fibre, as you know. So just this idea that we're meant to have 30 grams a day, but none of us do. We have something like, I don't know, an average of 18 grams. We're all kind of worried about our protein intake, but actually we're really deficient in fibre by about 30% every day. And when you're looking at these rural African diets, there was a guy in the 1960s, 90s, 70s called Dennis Burkett, who was this amazing doctor and epidemiologist, and he was looking at the amount of fibre that was eaten by rural Africans, and it was really well over 100 grams a day. And consequently, he looked into their gut transit time and stool consistency, which we know is an indicator of a good gut microbiome composition. And they were all incredibly healthy. And he felt it explained, before he even knew about the gut microbiome didn't exist then, obviously. Well, it did exist, but it wasn't known about in the 1970s. But he noticed that they had none of these Western diseases. So we're talking about everything from colon cancer to, cardiovascular disease, diabetes, obesity, thrombosis, vein problems, all these things. We know now it all makes sense in the light of what we know about the gut microbiome and what it actually does. So, and also just finally, sorry, Sheena, I'm just going to butt in slightly because I think it's really important about butyrate, which we love, and I call it beautiful butyrate because it's this amazing short-chain fatty acid. And in general, it is brilliant, anti-inflammatory and anti-cancer. But the thing is about the gut microbiome is nothing is always one thing or the other. And it's generally tumor suppressive, but there have been studies that demonstrate that it can, in very rare circumstances, sort of promote colorectal cancer, depending on the dose of the butyrate and the stage of the colorectal cancer. And they think it's to do with its impact on senescent cells, which I won't go into now. But just so in case listeners are reading something that says that butyrate isn't always brilliant and that there are circumstances in which it can be a problem, that's always going to be the case. I mean, and there's nothing in life which is ever 100% good or 100% bad, and it's all to do with the context. And I just think it's important. That's known as the butyrate paradox. So I just wanted to put that in there.
Okay, well, that's wonderful. I think we're going to wrap up for today. The next podcast, we're going to continue this journey into cancer and the microbiome. We're going to be talking about how the microbiome influences our cancer journey, so our treatments, and also our survivorship with cancer, and how we should be utilising our knowledge on human microbiomes to enhance that journey. So that's going to be the next one. And then we're going to follow that up with a really interesting podcast talking to a professor in oncology about microbiome science and cancer. So we're going to finish for today. Thanks very much for listening. I really hope you enjoyed this. Please check out the show notes if you want the references and if you want that very important recipe. And we'll speak to you next time. Take care. Bye.
Thank you so much for listening to this episode of Microbiome Medics Podcast. We really hope you enjoyed the content and we welcome your feedback. We'd love to hear any suggestions you might have for microbiome topics that you'd like us to cover, and we also appreciate listeners' questions and will endeavour to answer them in the next podcast.
So if you want to progress this learning further, look at the Gut Microbiome for Clinicians. It's an online course on bslm.org.uk, which will take you on a much deeper dive into the gut microbiome.